Ooh, fancy. They got brand new little thingies. Yeah. Am I too loud or am I good? Huh. Who knows? All right, sorry I'm late. Lab ran a little long today. How y'all doing? Tired? Okay, here's the good news. We won't go all day today. I'm only going to cover Chapter 8. How's Dr. Sarkar doing in class today? Okay, but your test only goes to 8 on Friday, right? So we'll get through 8. It'll probably take about an hour, and then we'll get you out of here. There is a mock test going around, so take that. It says Chapter 5 icosanoids on the front. They are divided up into sections, so you know where to look. So the first section is Chapter 5, and then after 10 to 20 questions, it'll change to Chapter 6. So it's not just a test over Chapter 5. Rolls going around, go ahead and sign that. Now, someone already brought up a question on question number 1. You guys can keep them, put them in your classroom somewhere. Someone already brought up a question on question number 1. Leukotrienes are synthesized directly from thromboxane A2, prostaglandin A2, 15 HPETE, or 5 HPETE. Remember, PGH2 is the intermediate before what? Before all the PGs and all the thromboxane. So it's not that. That's the cyclooxygenase pathway. So you're down to leukotrienes come from the lipoxygenase pathway, right? And all leukotrienes come from the lipoxygenase pathway. And what lipoxygenase synthesizes all the major leukotrienes, A, B, C, D, and E? Is it the 5 lipoxygenase pathway or the 15 lipoxygenase pathway? The 5 lipoxygenase pathway. The answer's 5, right? Yeah. So which is why that one says the intermediate directly before leukotrienes is 5 HPETE, because that comes out of the 5 lipoxygenase pathway. Now, the lipoxygenase pathway produces 2 things. It produces lipoxins and it produces leukotrienes. The leukotrienes come straight down the 5 pathway. The lipoxins do what? 15, then 5. So the answer would actually be the same if I'd asked what's the direct precursor of a lipoxin, and it would still be 5 HPETE, I think. He won't ask you that. But I just want you to get that the leukotrienes all come down the 5 lipoxygenase pathway. The lipoxins come down 15, then 5 lipoxygenase. Cool? Aside from the confused looks I'm getting, because many of you weren't at that tutoring and haven't yet watched that video, does anyone have any questions? 5 is icosanoids. There's no handout for that one. 6 is amino acids. There is a handout for that one. Did I give you guys the link yet? Okay. It's on your Facebook? So get on your Facebook. There's a link to the Google Drive that has all the Biochem stuff in it. Just bookmark it. You can give it to your friends. You can do whatever with it. It's got all the mock tests, and it's got all the stuff I've made for it. Not every chapter is there because not every chapter has the little sheet thingy that I made for it. Including Chapter 8. There's no little sheet thingy. There will be for Chapter 9, so eventually, well, no, I'll probably just keep printing them and bring them. But you'll see all the stuff on there, so if you want to get a jump, if you want anything for class, you're welcome to get it there. Any other questions? Anyone confused by COX blockers and why I spent a lot of time talking about COX in my last thing? Because apparently I got some amused comments from people that weren't here. We're starting in lecture number 8, which is the last lecture before your test, page number 88. Sorry, that is not lecture number 8. Let's start on page number 89, which is lecture 8. So lecture 9 is a lot of pathways. Lecture 9 is going to be a bunch of different pathways we're going to study. But the first thing we need to look at, which in lecture 9 will not be on your test, the first thing we need to look at is lecture 8. Lecture 8 has one common theme. Lecture 8 is the transfer of 1 carbon units. We're going to study 3 different things that transfer 1 carbon units. We're going to study when each of them transfers a 1 carbon unit and why they're going to transfer 1 carbon unit. That's it. These guys all juggle 1 carbon units. 1 carbon units can be methyls, they can be formals, they can be et cetera, et cetera, et cetera. C with anything attached to it, as long as it's not another C. It's a 1 carbon unit, right? So that's what these guys juggle. So very importantly, this chapter does not talk about biotin, which is highlighted in purple right there. That's the word biotin. Biotin is vitamin B7. B6 is pyridoxophosphate, B7 is biotin, B9 is folate. B7, I think. For some reason that's blanking in me right now. Pretty sure it's B7. But you don't need to know that for Dr. Sarkar. You will learn that in nutrition. We're not talking about biotin in this chapter. Biotin is not a 1 carbon transfer that we're talking about. But this is a very important test question. The most oxidized version of 1 carbon unit is transferred by biotin. The most oxidized version is carbon dioxide. It's a carbon and two oxygens. It's the most oxidized because it has the most oxygens, right? It is transferred by biotin. So when he asks you what the most oxidized version of one carbon unit is transferred by, it's biotin. It's not one of the three from this chapter. We're going to talk about the other things that are transferred by these guys in the order of oxidation and reduction. You're going to have to know that. But the most oxidized version of one carbon unit, the most oxidized one carbon unit is carbon dioxide, and it is always transferred by biotin, period. Please know that.
Transferred by or to?
By biotin. It is transferred to biotin, and then biotin gives it something else. Biotin is the coenzyme that does it. But all you need to know is biotin takes care of the most oxidized version. So one carbon groups at lower oxidation levels than CO2 will be transferred by the three guys we're going to talk about in this chapter. Those three are tetrahydrofolate, which is aka TH4 tetrahydrofolate, or FH4, which is a folate with 4 hydrogens attached, tetrahydrofolate. Vitamin B12, which we're going to talk about, and S-adenosylmethionine, aka SAM. Those are the three guys we're going to talk about. We start with tetrahydrofolate. Tetrahydrofolate, aka THF or FH4, is vitamin B9, folate B9, is vitamin B9. They transfer one carbon groups, which if it's any of these three, that's what you're going to answer on the test. What is the purpose of tetrahydrofolate? It transfers one carbon groups. THF is produced from the vitamin folate or folic acid. Please know that. That is the intermediate before we get tetrahydrofolate. That is the inactive form. So as we get into vitamins, you're going to learn about the active form and the inactive form. Folic acid or folate is the precursor or the inactive form. And when we get it to tetrahydrofolate, it's the active form. Now, you have a folate and you have a folate with 4 hydrogens attached. Which one is more reduced? A folate or a folate with 4 hydrogens attached? Folate with 4 hydrogens, because that was one of the rules. For reduction, we can add hydrogens. So how are we going to get from folate to folate with 4 hydrogens? We're going to reduce it, because it's more reduced than the other guy. So before we even get there, you know that already. So folate's a water-soluble B-complex vitamin. You might need to know that now. You will need to know that later. It's one of your B vitamins. All of your B vitamins are water-soluble. Might as well memorize that now. Every B vitamin is a water-soluble vitamin. It cannot be synthesized by humans. That means it cannot be synthesized within our body. I'm sorry, yes, within our cells, I should say. There are some bacteria that synthesize folate in your gut. It's one of those gut-synthesized bacteria that we're going to talk about later on. But it cannot be synthesized in a human cell, in a human system, which means it's a nutritionally essential vitamin. So I would know that. It's an essential vitamin in humans. It is obtained from the diet. Oh, there you go. Or intestinal microbes can synthesize it. So what is one of the ways, based on that, what are the 2 ways you can get a folate deficiency based on that? Lack in the diet, or your intestinal microbes aren't synthesizing it. Why wouldn't your intestinal microbes be not synthesizing it, for God's sakes? They're not there. Why aren't they there? Antibiotics. Nice job. Nice job. So those are things you need to know. He'll talk about them later in the vitamin chapter. You'll talk about them in nutrition. Anything that's synthesized in your gut by intestinal bacteria, if you take an antibiotic, will be synthesized less or not synthesized at all because it is an antibiotic. So please know your intestinal gut bacteria. This is the first one you're going to encounter. Folate is the doubly oxidized form of THF. We know that because THF is the reduced form of folate. That makes sense? The guy with the hydrogens is the reduced form. In this doubly oxidized form, folate can't transfer C1. In other words, it's inactive. Folate is the inactive form. And again, the folate versus folic acid, same thing as before. The 8 just means it's the conjugate base. It's deprotonated. Folic acid still has its proton. It's still an acid. So folate and folic acid are equivalent. One of them just has a proton and the other one doesn't. But to get from our inactive folic acid or folate to our active THF, we have to reduce it. It must be reduced to generate the active coenzyme. You must know the active form is THF, or FH4, or tetrahydrofolate, however you list it. All mean the same thing. Therefore, we have an enzyme that does this. It takes folate to dihydrofolate, and then dihydrofolate to tetrahydrofolate. Same enzyme, 2 different reactions. It's called dihydrofolate reductase. It's a reductase because it reduces. You need to know that enzyme. The enzyme that takes folate to tetrahydrofolate in 2 steps is dihydrofolate reductase. Just look for a reductase because you know folate is going to THF. It's being reduced. You know that because it's got more hydrogens on it. Now, I told you this before. This is one of your biochem pearls. All reductive biosynthetic processes primarily use what as a donor of a reducing agent? NADPH. All reductive biosynthetic processes generally use NADPH. Since this is a reductive process, since this is a biosynthetic process, because this part is happening in our body, we take in folate and we have an enzyme to convert it to the active form. We take in folate and then we have dihydrofolate reductase, which converts it to tetrahydrofolate. That enzyme is dihydrofolate reductase. It uses NADPH. Uses 2 NADPH, might be a test question, but I don't know on that one. 2 NADPH to go from folate to tetrahydrofolate, which makes sense, right? Because how many H's are associated with each NADPH? Not quite. Remember we showed you this before? When you have NAD and it goes to NADH plus H plus, you have FAD, which goes to FADH2, and you have NADP, I think plus, which goes to NADPH plus H plus. So each of them, from back in try 1, I don't know if I really hit that with you guys, carry 2 hydrogens. So if you need 2 hydrogens, or the electron equivalent of 2 hydrogens, you have to use 1 donor. Since this guy has 4 hydrogens, he needs 2 donors. So we use 2 NADPH. Where do we get NADPH? Pentose phosphate pathway, aka HMP shunt, hexose monophosphate shunt. What's the other place we get it? The malic enzyme reaction, which is part of fatty acid. Yes. Fatty acid breakdown. Biosynthesis. Fatty acid biosynthesis. Which is getting the acetyl groups out into the cytosol where we can use them. Thank you. Folate is mostly present in our body in its reduced form, which means it must be important, so most of the time we keep it reduced. Just a little trivia fact, not probably terribly important. Oxidation reduction. Okay, this is important. These C1 groups, here's your folate molecule right here. This is folate. This is actually tetrahydrofolate, sorry. These carbon groups are attached at one of 2 positions. They are attached either at the N5, which means the nitrogen at the 5th position, or they are attached at the N10, which means the nitrogen at the 10th position. Those are the only 2 places tetrahydrofolate can carry a 1 carbon group. Now, it can be attached to both. It can be linked to both at the same time, but the 1 carbon groups, TQ, are attached either to N10, to N5, or to N5 and N10. They're carried on one of the nitrogen groups, and you got to know that those nitrogen groups that they are carried on are the ones at position 5 and 10. That will show up, I almost guarantee. THF obtains its 1 carbon units from the following compounds, which means that this is a 1 carbon donor. What does it do? It donates its carbon, right? Which means it needs to refill that carbon if it's going to be able to donate it. So we have this usable pool of 1 carbon donors. You got to know this, they're called the 1 carbon pool. And these guys are the guys that THF can go over to and say, hey, I need a carbon. This guy over here, my buddy, he wants carbon. I need to get a carbon from you, and I'm going to take it over to him. He's a 1 carbon transfer, but he needs to come up with that carbon group from somewhere. The donors you need to know, the 1 carbon pool, are serine, glycine, histidine, formaldehyde, and formate. Histidine, formaldehyde, and formate. Please know those. Those are the 1 carbon pool, which means that THF can obtain a 1 carbon unit from any of those. THF can obtain a 1 carbon unit from any of those and attach it to itself at what positions? N5, N10, or both. Good. 1 carbon pool is a collection of C1 units attached to the THF, known as the 1 carbon pool, etc. Which means that he goes out here. There's this random thing. Here's serine. Here's the R, in this case, is not the side group. That's the whole amino acid. Here's the side chain of serine. And he takes that methyl group and ditches that hydroxyl group and turns it to this. So when THF steals the methyl group from here, what does it become? What is this amino acid? Just an amino acid plus an H. Glycine, right? So very important reaction you need to know. This is going to be one of the test questions. I think it's coming up in a page or so. That one of the things that THF does is it converts serine to glycine, and it can convert glycine to serine. So if you need to change these guys back and forth, this is part of the 1 carbon pool. It can take or donate a 1 carbon group to it. So THF converts serine to glycine. That's one of the places it gets its 1 carbon. So when THF converts serine to glycine, it gains 1 carbon. It becomes THF with a carbon attached to N5 or N10. It's ready to go donate it somewhere else. Questions on that? All right. Nice little complicated diagram. I'm going to kind of skim through it, show you some of the high points, but it's not something you're going to have to memorize. So what is this? What do we call this? That's the one carbon pool up at the top. Serine, glycine, histidine, formaldehyde, and formate. Tetrahydrofolate out here doesn't have a one carbon. I'm going to show it on the monitors for those playing the home game. Tetrahydrofolate out here does not have a one carbon unit. It slides in, grabs one carbon from its one carbon pool, becomes tetrahydrofolate with one carbon, and then comes down here and turns a precursor into a product. Cool? Now, while this one carbon is attached to it, it can be oxidized and reduced. Now, this is that interconversion. These things can happen while it's attached to it. So over here, you just have plain old tetrahydrofolate. It picks up something, a formal group at N10, and then it converts it to a methanol group at N5, N10, where it's attached to both, and then converts it to a methylene attached to both, and then a methyl attached to C5. So it can juggle this guy while it's attached to it. It can be oxidized, it can be reduced, it can be moved around while it's attached to it. That's all you need to get from that. The one carbon units can be oxidized, reduced, or juggled around while attached to the THF. Once they're attached, they're not pure, that's what they are. They can be moved and changed a little bit. And then he's going to go down here and donate something to a precursor, turn it into a product, and here's some of the products it does. It makes serine. What does it donate a one carbon group to make serine? What does it donate? Glycine, right? It takes glycine and turns it right back into serine. It makes DTMP, which we're going to talk about. It makes purine bases, which we'll talk about in Chapter 9. And it makes B12 attached to CH3. Now, at the beginning of this chapter, I told you that B12 was a one carbon carrier, right? Where does B12 get its one carbon unit from? Who gives the one carbon unit to B12? THF. THF donates a precursor to make B12. B12 methyl right here. So what's the one carbon pool for THF? Serine, glycine, histidine, formaldehyde, formate. What's the one carbon pool for B12? THF. Nice. Exactly. So THF serves an important role in donating one carbon group to something, which might be a methylation reaction, but it also serves an important role in methylating another methylator. It donates the one carbon group to B12 so that B12 can go and donate stuff to other guys, because B12 is also a one carbon donor. So that means, we'll talk about that later. Okay. So C1U groups can be attached at N5 or N10 or both. That's your TQ. Make sure you know that. They can be juggled and moved around. They can be oxidized or reduced or moved while they remain attached, so they're not just stable. Now, this chart's going to be on both places. I want you to write this in because you got to know this. The most oxidized form of C1 is transferred by who? Biotin. Nice. What's the most oxidized form of CO2? Good. But the most oxidized form that THF carries is formate. Most oxidized up here, has the most oxygens compared to hydrogens. Please know the oxidation reduction. Most oxidized is formate, and mainly you just have to know the ends. Most reduced on this chart is methanol. That's not entirely true. What's more reduced than methanol? What can you add at the bottom there? What's a C with no O's attached to it? Might have 3 H's. Methyl group. So you could add at the bottom of that chart a methyl group. But the most oxidized form is formate, then formaldehyde, then methanol, then methyl. Trust me, some of the questions we'll ask, it'll show you N5, methanol, N10, whatever, and it'll say, is this more oxidized or less oxidized than this? So you need to know the order of oxidation of these products. Now, he's given you a nice little chart over here on the top of page 93. Oxidation level methanol, there's a methyl. Methyl should technically be above it. It's going to show you N5-methyl THF, which is more reduced than N5-N10 methylene, which is more reduced than formal, which comes from a formate. So just make sure you know that methyls and methanols are more reduced than formaldehydes, which are more reduced than formates. Please know that order. However you want to learn, oxidation-wise or reduction-wise, know the order, because you will get a test question about that. But the most oxidized form is what? Most oxidized form of a one carbon unit. CO2. Who carries it? Biotin. Good. The most oxidized form carried by THF is a methanol. Is a formate. Okay. Questions on anything we've talked about so far? No? Okay. We're going to take a little dip off the deep end now. We're going to talk about what it participates in. THF participates in the transfer of one carbon units. Hooray, we know that. THF is responsible for the conversion of deoxyuridine monophosphate, all you need to know is dump, to deoxythymidylane, which is DTMP, by thymidylate synthase. Now, where have you seen U's and T's before? Okay, you might not, oh yeah, you might have seen them in RNA. You might have seen them in RNA, which is kind of weird because you don't really use the deoxy version in RNA, but it is required for the conversion. So we're going to talk about Chapter 9, the synthesis of purines and pyrimidines. So you're going to see this in detail in actually you're going to see this in less detail in Chapter 9, but you need to know it now. This is important for the creation of the T ribonucleotide. This is important for the creation of the thymidylate, which we need for what? RNA or DNA? DNA. A TGC is DNA, a UGC in RNA. So if we need a T, we're making DNA, which is one of the reasons when you study it, and I'm sure you've heard this before, what's one of the most important vitamins for a pregnant woman? Folic acid or folate, because it's important for DNA synthesis. And when we study folic acid, one of the big things you're going to see is neural tube defects and mental retardation and all this stuff that deals with the nervous system. Because what's the first system formed and what needs a lot of DNA? The nervous system. So one of the big things that folic acid is involved in is the creation of the thymine or thymidylate, however you want to call it, whether it's attached to phosphates or not, thymine or thymidylate DNA version of the T so that we can make DNA. So this guy up here takes DUMP, which is the precursor, which we're going to synthesize next chapter, and he converts it to DTMP by doing what? What does folate do? What does tetrahydrofolate do? Methylates stuff. It donates a one carbon group. If he asks you anything, how does tetrahydrofolate convert DUMP to DTMP? Methylates it. How does it convert glycine to serine? It methylates it. How does it convert YETCF? It methylates it. Or, sorry, it adds or subtracts a one carbon group. In this case, we're adding a methyl group. So the methylation of DUMP turns it into DTMP. Really easy enzyme, thymidylate synthase. It's telling you what it's making, thymidylate synthase. This is a thymine ribonucleotide base. The reason it's called thymidylate is because it's attached to phosphates. When it's just a T, it's called thymine. When it's a TMP or a TDP or a TTP, with how many phosphates attached to it, it's thymidylate. You have uracil and urolate, adenosine and adenylate, et cetera, et cetera. I confuse anyone on that? Okay. Thymidylate synthase makes the T base. In the process, you go from a methylated or a one carbon carrying THF or FH4. It's carrying it at the N5 and N10 position. It's going to roll through this. It's going to donate the methyl group, and it's just going to become dihydrofolate. Is dihydrofolate the active version? No. What enzyme will take dihydrofolate to tetrahydrofolate? Dihydrofolate reductase. What does it need? What needs NADPH? Fantastic. Takes it back to FH4. What's it doing here? It's getting remethylated from its one carbon pool so it can continue to do this again. So every time it comes up here, it donates its one carbon group. I'm not sure the specifics of when it gets oxidized, when it doesn't, but sometimes it will. And then it needs to be reduced again so that it can pick up another one carbon unit so that it can do it again. That make sense? So that's the cyclical nature why we can keep making thymidylate bases for our DNA synthesis. Now, a couple of blockers you need to know, and that's all you need to know. What you need to know is that folate is important for, it's an important cofactor for thymidylate synthase. It's important in the creation of the T base for
This is the security department.
DNA.
Please be advised that the creek lot will close at 6 p.m. Please move your vehicle from the creek lot to the main campus at this time. Thank you.
I'll say that again. Folate is important for the synthesis of DNA. It's an important coenzyme, actually, for thymidylate synthase, and it donates the one carbon group for the creation of the thymine base in DNA. Cool? Now, this process can be shut down in two places. You need to know the two places it can be shut down. It can be shut down by 5-fluorouracil, which blocks the enzyme thymidylate synthase. The blocker 5-fluorouracil blocks the enzyme thymidylate synthase. Right down here, blocker of thymidylate synthase. Methotrexate shuts down dihydrofolate reductase. Methotrexate shuts down dihydrofolate reductase. because it's a structural analog of folate. Who wants to take a guess at what type of inhibition methotrexate does to that enzyme, dihydrofolate reductase? It can be allosteric, and if it's allosteric, it can be competitive or non-competitive. Which is it? Competitive. Why? Because it looks the same. Because it's a structural analog. Anything that's a structural analog means it's going to bind to the same place. I mean, there's an off chance it's not. But anytime they tell you it's a structural analog, what that means is it looks really similar. It shares a lot of the same similarities. So what it's going to do, it's going to bind in the active site. The more of it there is around, the more dihydrofolate reductase you're going to stop. Because all it does is it takes the place of folate or difolate in the active site of the enzyme. So it's a competitive inhibitor. It's not an allosteric inhibitor, because allosteric implies other site. Allosteric implies non-competitive. So it's a competitive inhibitor because it has the same structure. Anything that's structural analog is likely a competitive inhibitor. So folate, important for DNA synthesis, forms the T base through thymidylate synthase, which can be shut down by 5-fluorouracil. Or we can not be able to make the active form of folate because dihydrofolate reductase is being competed with by methotrexate. Questions? Good news is this is the most complicated one in this chapter. And I think we're at the end here. So here's this nice little complicated diagram which shows lots of stuff that you probably aren't going to want to memorize. So here's serine and glycine donating their 1-carbon units, forming methylated FH4, which is coming down here, and it's contributing through B12 to this formation, which we're going to talk about. That's all in the rest of the chapter. Or it can come over here, and it can convert methylene. Yeah, don't worry about it. Don't worry about that page. It doesn't make a lot of sense. Questions on folate before we move on to B12. Like I said, that was the worst guy in this chapter. Need to know the active form, the inactive form, the enzyme responsible for its creation, the coenzyme that enzyme requires, which is what? What's the coenzyme that dihydrofolate reductase requires? NADPH. Nice. It's a coenzyme, right? It's an organic substance. And you need to know what it's important for, which is what? Big picture. What's it important for? DNA synthesis, because of it makes the T nucleotide. It's the only way you can get the T nucleotide. Make sure you own that chart on the last page. You're going to be asked about both, not the structures, but what it converts, what the enzyme is, and what the blockers are of it. All right. What does B12 do? Transfers 1 carbons. Where does it get its 1 carbons? From THF. Perfect. So we've already talked about half of this, guys. B12, very importantly, meat, eggs, dairy proteins, fish, poultry. What do you not see in that list? Vegetables, fruits, plants of any kind. B12 only comes from animal proteins. Vegetarians usually don't have trouble with this. They'll get it in certain supplements and stuff, so it's not a big deal, but vegetarians who don't know what they're doing can often be B12 deficient. And B12 deficiency is very bad neurologically. B12 is really important for the nervous system. So B12 is all meat products, all animal products, I should say. It's not synthesized in higher animals such as humans, just in lower animals. So how do higher animals such as humans get it? By eating lower animals, right? So we get it from other stuff that makes B12, but it's an animal product only. It's not synthesized in plants. That's a big point for nutrition and for any patients you may encounter. The structure of vitamin B12 contains a corin group, which is similar to a porphyrin ring. Where do we see porphyrin rings? Did we talk about that yet? No, it's chapter 9. He talked about it in class, probably. That's in heme synthesis. Okay, that's next chapter. We're going to talk about porphyrin rings. Sorry, I'm getting ahead of you guys. Porphyrin ring. So the structure of B12 looks almost identical to a heme group. The only difference is the ion in the middle. The ion in the middle in B12 is cobalt, which is why the AKA for vitamin B12 is cobalamin. Cobalamin. What's the ion in the middle of a heme group? Fe, iron. Good. So vitamin B12 contains a corin group in the middle, which is similar to porphyrin, except it contains cobalt. Please know that the center ion in B12 is cobalt. So what do you think is important for the synthesis of B12? Cobalt. Good. Fantastic. If you care, oh, there you go. It says it right at the top. Vitamin B12, a.k.a. cobalamin. Come chapter 10. You're going to have to know all of those. B9 equals folate, B12 equals cobalamin, etc. So if you're getting on it now, you're ahead of the game. Transfer, it's responsible for two things. So we had two good examples from folate. What were the two good examples? What are the two reactions we care about from folate? What's the one we talked about? What's folate important for? DNA synthesis, because it transfers a one-carbon group from DUMP to DTMP. What's the other one? Where does it get the one-carbon unit that we used at the example at the beginning of the chapter? Serine to glycine. So if he asks you example things on the test and he says, which one of these is responsible for the conversion of serine to glycine, you're going to pick folate. Which one is responsible for the conversion of DUMP to DTMP? Fantastic. Whereas this guy, you have to know two different reactions. For this guy, you have to know homocysteine to methionine and methmalonyl-CoA to succinyl-CoA. Those are the two example reactions you need to know for B12. So he can put up one of these example reactions on there and all three of B12, whatever, and you need to know which one it is. So this guy can rearrange, it can transfer a one-carbon unit from homocysteine to methionine. What does homocysteine look a lot like? Cysteine. What's the difference between cysteine and methionine? Do you guys remember the structure? Cysteine ended in that terminal sulfhydryl group, whereas methionine has an extra methyl group on the end of that terminal sulfhydryl group. I'll point that out one more time. So if you look back at the amino acid structure, they're both sulfur-containing amino acids, but cysteine ends in a terminal sulfhydryl group, which is why cysteine can make what types of bonds? Disulfide linkages. Nice job. Which is why cysteine can make disulfide linkages. Methionine can't, because methionine has a methyl group attached to the end of it. So how do we get cysteine to methionine? We methylate it. Who methylates it? Vitamin B12, a.k.a. cobalamin, because it contains a cobalt. Fantastic. So vitamin B12 has its methyl group. It donates its methyl group to homocysteine to make methionine, and it doesn't have a methyl group. Where does vitamin B12 get its methyl group from? Where does vitamin B12 get its methyl group from? THF, which is why I drew right over here that THF attached to a CH3 comes on over and donates it, and then you could draw a little THF coming off over here, because its donor of one carbon group is THF. So tetrahydrofolate cannot directly donate a methyl group to homocysteine to make methionine. Tetrahydrofolate cannot directly donate a methyl group from homocysteine to homocysteine to make methionine. It has to act through B12. So the way it acts through B12 is by giving B12 a methyl group so that B12 can directly donate a methyl group from homocysteine to methionine. The other one it does is methmalonyl-CoA to succinyl-CoA. The only place you're going to see this, and we're going to kind of make a connection because it happens over a couple different chapters. What's succinyl-CoA? Intermediate of the TCA cycle. Methmalonyl-CoA is an intermediate in the conversion of propionyl-CoA to succinyl-CoA. Methmalonyl is an intermediate in the conversion of propionyl-CoA to succinyl-CoA. Where do you see propionyl-CoA? The oxidation of odd chain fatty acids. Remember, when you oxidize an odd chain fatty acid, you end up with acetyl-CoA and propionyl-CoA, which is, guess what? A 3-carbon unit. Propionyl-CoA will be converted to methmalonyl-CoA, and then methmalonyl-CoA will be converted to succinyl-CoA. So B12 is very important in the breakdown of odd chain fatty acids. Sorry, B12 is very important in the breakdown of odd chain fatty acids. I don't think he'll go that far in your test, but don't know. B12 is very important in the breakdown of odd chain fatty acids. Now, it's not truly a methyl donation, it's just a rearrangement. So it picks it up from one area and it donates it to another area. Again, don't think that's high priority for you to know, but B12 is important in the conversion of methmalonyl-CoA to succinyl-CoA and homocysteine to methionine. Have I drilled that enough? Yes. Your deadpan looks say yes. Last guy of the chapter. You guys are almost out of here. I know, really short day today. S-adenosylmethionine. SAM is a methylating agent in several reactions that are not favorable from THF. What does that mean? If THF can't do it, SAM takes over. If THF can't do it, B12 takes over. These guys cover for when B12 can't do it. B12 is your primary one-carbon transfer. But if they can't do it, you've got to rely on someone else. So again, SAM takes over in a couple of different places. SAM adds CH3 groups to either oxygen or nitrogen. I would note that SAM will only add them to an oxygen or a nitrogen. SAM will only add them to an oxygen or a nitrogen. Synthesis of SAM and its breakdown is synthesized from the reaction of ATP and methionine. We're going to look at that, but what's that called? SAM is S-adenosyl methionine. S-adenosyl methionine. What does that mean? It means it's a methionine with an adenosyl attached through the S group. What's the S group? Sulfur. And does methionine have a sulfur? So SAM is made from adenosine and methionine, and adenosine is ATP. SAM is synthesized from the reaction of ATP and methionine combining together to make S-adenosyl methionine. So you must have ATP present, you must have methionine present. What else is methionine important for? Nope. Start codon. Methionine is the start codon in all your proteins. Synthesized from the reaction of ATP and methionine. As with activation of vitamin B12, ATP donates. Yeah, that's fine. So, final look at this chart. Almost final. Over here, what's this? Tetrahydrofolate attached to a methyl group. Who's it donating its methyl group to? B12. Who's B12 donating its methyl group to? Homocysteine to make methionine. Methionine is then picking up an adenosine from this ATP, becoming S-adenosyl methionine, aka SAM. SAM will then lose its methyl group, becoming homocysteine, by donating it from a precursor to a methylated product. Does that make sense? So, tetrahydrofolate gets a methyl group. Tetrahydrofolate gives its methyl group to B12. B12 gives its methyl group to homocysteine. Homocysteine becomes methionine. Methionine becomes SAM. SAM donates its methyl group, and methionine goes back from methionine to homocysteine because the difference between methionine and cysteine is that terminal one-carbon group. So SAM donates its one-carbon group, goes back to SAH, S-adenosyl homocysteine, then it ditches the adenosine, it goes back to homocysteine, it goes through the cycle all over again. The only reason we do this is because these are reactions in which THF cannot donate a one-carbon group. And we're going to look at them on the next page. Do you get that diagram? Okay. Questions? Yes. From methionine, we go to S-adenosyl methionine because the S-adenosyl methionine is methionine plus adenosyl, which is ATP. Adenosine, which is part of ATP, that's the A. So as you can see here, the ATP is coming in, and all that's leaving is the 3-phosphate. So what stayed? The A. What's the A? Adenosine, which is where you get the adenosyl methionine. So it's actually picking up the adenosine group from the ATP to become SAM. And then when SAM donates a methyl, it becomes SAH, S-adenosyl homocysteine, by methylating a product and then continues on. There are more? This can only happen when THF is working, but it only happens in reactions that THF is not favored. In other words, THF is going to donate the methyl group everywhere it can. If it can't donate it, B12 is going to donate it. If B12 can't donate it, SAM is going to donate it. If B12 is not, or I'm sorry, if folate is not working, what shuts down? This whole thing. Everything shuts down. So it's actually a big clinical picture, and they're going to drill that in clinical nutrition, is a lot of people will come in with B12 deficiency symptoms, which are neurological symptoms. You get a B12 deficiency, your nerves start to demyelinate peripherally, and your spinal cord starts to demyelinate. That's bad. So what do? They're like, oh, their nerves are demyelinating, they need B12. They give them B12, they don't get better. Why? B12 is fine. It's folate that's deficient. And what's the primary source of folate? You'll learn in nutrition is green leafy vegetables. What do Americans notoriously not eat? Green leafy vegetables, because they taste like crap, right? So nobody eats these green leafy vegetables. They get all their stuff from vitamins, and not a lot of vitamins have folate. So you will actually see B12 deficiency symptoms from folate deficiency, because if folate isn't working, B12 isn't working, period. Another question? Cool. Interesting clinical stuff to know. And actually the good thing, and we're going to talk about this in the water-soluble vitamins chapter, is what's the harm in administering a water-soluble vitamin? There is none. It's water-soluble. It's excreted freely. There are a couple that are stored for a little while, But primarily, you can take a toxic dose. It takes a ton of effort to take a toxic dose. But water-soluble vitamins, you can pretty much freely administer. So how do you check if they're B12 or folate deficient? You give them B12 and folate and see if they get better. Fat-soluble vitamins are a different story because they're not excreted freely because they're fat-soluble, not water-soluble. Now, the final things for this chapter, and you need to know this, I've kind of added everything to the chart. This makes it complete. I don't know why this is in this chapter because you're going to talk about this next chapter, but you do need to know it just in case he asks it on the test. Important reactions that SAM is a part of, and the ones I want you to know are the top three. Norepinephrine to epinephrine, guanidinoacetate to creatine, and nucleotides to methylated nucleotides. We're going to talk about all three of those next chapter. SAM is very important for epinephrine synthesis, creatine synthesis, and methylation of nucleotides. Please know that for your test. And like I said, we're going to go through all three of those pathways. You've actually probably already talked about creatine synthesis, maybe. It's near the beginning of chapter 9. So if you haven't talked about it yet, you'll get to it maybe this week or next week, or I'll talk about it next Monday. SAM is very important for creatine synthesis, epinephrine synthesis, and methylated nucleotides. I put your other four examples here that you need to know. Conversion of DUMP to DTMP for DNA synthesis is THF. Conversion of serine to glycine is THF. Conversion of homocysteine to methionine is B12, and methmalonic acid to succinyl-CoA is B12. I know you guys really want to methmalonic acid, methmalonyl-CoA. Methmalonyl-CoA, methmalonate, or methmalonic acid are all the same thing. To succinyl-CoA. Now, I know you guys are very excited. It's 6 o'clock. You have nothing going this week because I'm sure it's not midterms for you. I really don't think we should continue on into Chapter 9 since you guys are lecturing, since you guys have a test, and I have plenty of time. Let me check before I let you go. Well, you may not have a choice. I get to dictate that. So your test is this week. I have 1, 2, 3, 4. Yeah, I got plenty of time with you. So we are done. Feel free to stay and ask some questions or get out of here and study. You have the link somewhere to all the information I've posted. Get it from a friend. Thank you. Get it from a friend if you don't have it. It has all the study guides if you're missing any. It has the mock exams if you want to print extras or if someone didn't get them. Next week we will be blowing right on through Chapter 9, which is a big chapter. Chapters 9 and 10 are both very big chapters, so come prepared to get your mind exploded a little bit next chapter because we're going to go through a ton of pathways. But today you are free. Get out of here, study hard, and I like you guys.